Hi guys, so we're going to be going over chapter 5, which is titled The Structure and Function of Large Biological Molecules, and we can also call our large biological molecules macromolecules. And the prefix macro translates directly to large or big. And we have four main classes of macromolecules that make up everything that is living. And they include our carbs, so our carbohydrates. It includes proteins, nucleic acids, and lipids. So the first thing that we're going to talk about is polymers. And essentially, a polymer is just a long molecule that is going to consist of repeated units, and those repeated units are going to be called monomers. And something important to note is that polymers, they are linked by covalent bonds. So when we have monomers that come together to form a polymer, they are linked by covalent bonds. And our polymers, they're essential for life. They form the structural and functional components of cells. And when we have large polymers, that's when they become known as macromolecules. And macromolecules, they're known for their size and for their complexity. And they're going to carry out a variety of function that we see in living organisms. So we have four groups of macromolecules, which include carbohydrates. And we can think of carbohydrates as our sugars. We have proteins, and we in our protein groups we have amino acids, and our proteins, they're going to be made up of amino acids. So it's kind of like amino acids are proteins monomers, and then we have our nucleic acids. And our nucleic acids include our DNA and RNA, which are both made up of nucleotide monomers. And their function is to store and transmit genetic information, which means that they have an incredibly important role in protein synthesis. So in carbohydrates, we have sugars, monomers of sugars that come together to form more complex carbs. In proteins, we have amino acid monomers. And in nucleic acids, we have nucleotide monomers. And when we look at lipids, we see that they are not a true polymer. And they are not true polymers because they are not made up of repeating monomer units like we see in our carbs, proteins, and nucleic acids. Lipids, instead, they are actually classified as polymers because they are large and they are assembled from smaller subunits. And those subunits include fatty acids and glycerol. And a lipid's function is for the formation of cell membranes, signaling molecules, and they are extremely important for the storage of energy. So, how are polymers synthesized and how are they broken down? Well, the chemical processes that our cells are going to use to make and break polymers are the same in all types of macromolecules. And these processes, they occur through the action of enzymes, which we know are proteins, and proteins are macromolecules. And enzymes are specialized macromolecules that are going to speed up chemical reactions within cells without being used. So enzymes are not used up when they carry out their functions. So I previously said that polymers, they are held together by covalent bonds. And monomers are connected by covalent bonds making up our polymers. And those covalent bonds that hold our polymers together, they are formed through a process that is called dehydration reaction. And in dehydration reaction, we have the loss of a water molecule. And that loss of a water molecule is what's going to result in the formation of a covalent bond. So essentially, what happens is that we have the hydroxyl end of a polymer, and we have the hydrogen of a monomer. And these two ends, they're going to come together to form water, and that water is going to be released out into the environment. And when we have this release of the water molecule, our monomer and our polymer, they form a longer polymer right here through a covalent bond. And we always have to note that whenever a reaction is carried out, a cell must invest energy for that to happen. And this investment of energy is what's going to allow our cells to create complex molecules, which include our macromolecules. And it's extremely important that we have the ability to break down our polymers, because a lot of the times our cells, they cannot actually absorb polymers because they are too large. So the covalent bonds that link our monomers, they are disassembled through hydrolysis. And we can think of hydrolysis literally as the opposite of dehydration reaction. So in hydrolysis, our covalent bonds, they are broken because of the addition of a water molecule. And what happens is that here we have a polymer that's just kind of floating around, and we have water all around the environment. And when this water molecule, when it attaches to the polymer, the hydrogen atom from the water molecule is going to attach to one of the monomers. And say it decided to attach to this one. So the hydroxyl group of water is going to attach to the other monomer. And once they attach, we're going to have the bond be broken, which is why we see a polymer and a monomer. And this happened because of the addition of water. Because a hydrogen atom from a neighboring water molecule attached to one of its monomers, and the hydroxyl group of that water molecule attached to another monomer. And because it attached, it broke our polymer down into a polymer and a monomer. So once we have the monomers in our cells, they're going to make new polymers through dehydration reaction. And that formation of new polymers is what's going to be tailored to the specific needs of each type of cell. And a common example of hydrolysis is digestion. And it's so important because when we take in food as organic polymers, they are too large for our cells to be able to absorb. And in our digestive tract, enzymes, they're going to direct hydrolysis of specific polymers. And the monomers that resulted from those polymers, it's going to be absorbed by the cells that line our gut, and it's going to be transported to our bloodstream, where it's going to be distributed to other cells. And the cells of our bodies, they're going to use dehydration reaction to assemble the monomers into new polymers, new and different polymers. And then they are going to carry out functions that are specific to the particular cell type that they were ingested by. So it's important to note that we have a very large variety of polymers, and all of these polymers, they actually come from only 40 to 50 different monomers. So each cell has thousands of different kinds of macromolecules, and all of these macromolecules, they have a variety of diversity, and this diversity increases in certain cases. So the varying diversity of macromolecules among the cells of the same organism, of the same individual. So the diversity found in macromolecules only in my cells is great. But this diversity is going to increase when we compare it to another human. So the diversity between my own macromolecules is great, but the diversity between mine and yours is even greater. And this diversity is going to increase when we compare the macromolecules of different species. And this diversity is going to come from 40 to 50 different common monomers. And we do have some that occur rarely. So we can think of our monomers kind of like our alphabet, right? So we have 26 letters in the alphabet, and those 26 letters, they come together to create a whole dictionary of words. They create a whole language. And those 26 letters, we can think of them like our monomers, and we can think of our words like our polymers. And it's important that we realize that all small molecules that are common to organisms, they are ordered into unique macromolecules. And even though we have a large diversity between organic molecules, the members of each of the 4 major classes of macromolecules, they are still similar in structure and function. So now we're going to talk about carbohydrates. And carbohydrates, they include sugars and their polymers. They are necessary for energy, and they also function as structural components. And we have 3 different kinds of carbohydrates. We have monosaccharides, which is our simple sugars. They're called our simple carbohydrates, and a monosaccharide example is glucose. We have disaccharides, which is 2 sugars that are covalently bonded, and that is an example of sucrose. So we can kind of think of disaccharides as double sugars. And the last kind that we have are polysaccharides, which are many monosaccharide polymers joined together. And this many just means more than 10 monosaccharides. So our sugars, our smallest carbohydrates, they serve as fuel and as a source of carbon. So our monosaccharides, they serve as fuel and carbon source. They are the building blocks of carbohydrates, and normally they're going to follow this formula, CH2O to the N power. And a common example of this is glucose. And the formula of glucose is C6H12O6. So if we were to replace our N with a 6, we see that we get C6H12 and O6. And structurally, our monosaccharides, they have 2 key functional groups. They're going to have a carbonyl group, which is what we see here in pink, and they're also that carbonyl group is going to determine if we have an aldose, so an aldehyde sugar, or a ketose, a ketone sugar. And monosaccharides, they also have multiple hydroxyl groups, and these hydroxyl groups are going to make them highly reactive and versatile. In glucose, we see that we have 5. 1, 2, 3, 4, and 5. And we can also categorize monosaccharides by the size of their carbon skeletons. So we have our hexoses, which have 6 carbons, which is glucose. We see 6, and if we count it, we have 6. 1, 2, 3, 4, 5, 6. We have pentose, which has 5 sugars, and we have triose, which has 3 carbons. Pentose has 5 carbons. So it's really important to know that sugars, they are the smallest carbohydrates, and they serve as fuel and a source of carbon. So monosaccharides, they serve as fuel for ourselves and our source of carbon. We need to know this. This is very important. But interestingly enough, sugars, they have the ability to differ in the spatial arrangement of atoms around asymmetric carbons, and they create isomers. So if we remember, isomers are the same molecular formula, but with a different structure. And we frequently see this in glucose and galactose. So when we look at glucose, we see that we have 4 hydroxyl groups on the right side, and we have 1 on the left side. But when we look at galactose, we have 2 hydroxyl groups on the right and 2 on the left. So even though they have the same exact formula, which is C6H12O6, they are two different molecules because they are isomers of each other. And although we normally see our molecules drawn in this linear skeleton, when we have our molecules in aqueous solutions, they form rings a lot of the time. And this is just what the ring conformation would look like. And this is a glucose because we see that we have 4 hydroxyls on the right and 1 on the left. So functionally, monosaccharides, they have a critical role in energy sources. So glucose, it is a major fuel for cellular work, and energy is extracted through cellular respiration. And we are going to have a whole chapter dedicated only to cellular respiration, which is chapter 8. But the other function of monosaccharides is that they serve as raw materials. They serve as building materials. So our simple sugars, they're going to be used to synthesize other molecules like amino acids and fatty acids. And two monosaccharides, they have the ability to join through glycosidic linkages. And these glycosidic linkages, they form through dehydration reaction. And when they are bonded together, they form our disaccharides, our double sugars. And examples of disaccharides include maltose, which is 2 glucose molecules coming together. Sucrose, which is glucose and fructose coming together. And sucrose is so important because it's used by plants to transport sugars. And we also have lactose, which is glucose and galactose joined together. And that is the sugar that we find in our milk. So we mentioned that polysaccharides, they are made up of more than 10 monosaccharides. But even though we use the number 10 as our defining point of disaccharide to polysaccharide, polysaccharides are made up of hundreds to thousands of monosaccharides, and they also are joined by glycosidic linkages. And they play an important role in storage and structure within living organisms. So essentially, our polysaccharides, they are just long chains of monosaccharides that are linked together by glycosidic bonds, and they have 2 primary functions, although they have a variety of other functions. So some polysaccharides, they are important for storage, and they are hydrolyzed as our sugars need them, as sugars are needed by our bodies. And the other main function that they have is to serve as building materials for the entire cell or whole organism. They are the building materials of our bodies. And the way that the specific role of a polysaccharide is decided, it depends on the sugar component and how the glycosidic linkages are arranged. So an example of a storage polysaccharide is starch. And we hear the word starch a lot, especially when we are talking about potatoes, but they are mainly found in grains also, in addition to potatoes. And starch, it is an example of a storage polysaccharide that is composed of alpha glucose monomers. So it's made up entirely of glucose monomers, specifically alpha glucose monomers. So, for example, our plants, they store their excess glucose as starch in structures that we call plastids, and plastids include chloroplasts. And when plants need energy, they break down that stored starch and turn it into glucose. And that's what I mean by our starches are some of our monosaccharides, some of our carbohydrates, they are hydrolyzed as needed by our bodies. So animals, especially herbivores, they have enzymes that are able to break down starch into glucose, which is what makes it a really, really important nutrient source. And starch specifically is going to consist of glucose monomers that are linked by 1 to 4 glycosidic linkages. Its simplest form is amylose, which is what we have here, a straight chain, and we have amylopectin, which is branched, and what we see here. So its simplest form is amylose, it's unbranched, and there are more complex forms like amylopectin that are branched. So another storage polysaccharide that we have is actually called glycogen. And glycogen is the storage equivalent of starch in animals. So starch is found in plants, and glycogen is found in animals. We need to understand that distinction. And glycogen, it is extremely branched. Look at all the branches that it has. It's just really branched. So it's a more branched version of amylopectin. And our glycogen, it's going to be stored in our liver and our muscles. And it's also going to be hydrolyzed as needed. But our glycogen reserves, it only lasts about a day. So glycogen is only able to provide a short-term energy reserve of 1 day. Another structural polysaccharide that we have is called cellulose. So cellulose is a key component of plant cell walls. And plants, they produce an immense amount of cellulose. They produce like 100 billion tons every single year, and I'm not exaggerating. But because they produce so much cellulose, it's going to make it the most abundant organic compound on Earth. And cellulose and starch, they are both polymers of glucose. But I did mention that starch is formed from alpha glucose monomers. Meanwhile, cellulose is composed of beta glucose molecules, and this composition is going to create straight and rigid structures. So, again, starch is made of alpha glucose, which is able to form helical structures, and cellulose, on the other hand, it's made of beta glucose. And in this structure, every other glucose molecule is flipped upside down, which creates a straight chain, which is what we see here. So these straight cellulose chains, they're going to form hydrogen bonds with each other, and they're going to make strong microfibrils. And these microfibrils, they are the building blocks of wood, paper, and cotton. And something that's a little bit of a fun fact is that humans, we cannot digest cellulose, but we can digest starch. And that's because the digestive enzymes that we have, they have the ability to break down alpha linkages in starch, but not the beta linkages that we see in cellulose. So as a result, our cellulose is going to just directly pass through our digestive system in the form of insoluble fiber. And that's actually going to help our digestion stay regular. But on the other hand, certain microbes, they have the ability to digest cellulose, and they do this through the enzyme called cellulase. And many herbivores, like cows and termites, they depend on symbiotic relationships between their microbes to be able to digest cellulose. So this ability to digest cellulose is going to give them access to cellulose as an energy source. And certain fungi are also able to digest cellulose. So now we're going to talk about lipids. And lipids are a really diverse group of hydrophobic molecules. So we know that lipids, they do not form polymers. And that is why they are not considered true macromolecules. However, lipids are composed of smaller units, and they are also large in size, so they become classified as a macromolecule. And what makes a lipid unique, or the defining characteristic of lipids, is the fact that they are hydrophobic molecules, meaning that they do not mix with water. So the reason that they are hydrophobic is because of their hydrocarbons. And we know that hydrocarbons, they are going to form nonpolar covalent bonds. And the hydrophobicity that lipids demonstrate, that's going to exemplify the structural role they have in forming the phospholipid bilayer. And the phospholipid bilayer, it's a membrane that's going to stop water from passing through, and it just serves as a barrier for cells. And these lipids, they are extremely diverse in their form and their function, and they have extremely important roles in our energy storage, the formation of cellular structures, and even in signaling. So a lot of the times, our lipids, they're referred to as fats. Fats also are not polymers, but they are large molecules that are assembled from smaller components via dehydration reactions, which is the same way that monomers come together to form polymers. So fats, they are actually made up of two parts. Glycerol, which is a small molecule. It's a 3-carbon alcohol that's going to have a hydroxyl group attached to each carbon. And this is what glycerol looks like. We have our 3 carbons, and we have our hydroxyl groups attached to each of them. And then we have all of our hydrogens as well. So this is the structure of glycerol. And the next thing that fats are made up of are fatty acids. And fatty acids, they are long hydrocarbon chains that have a carboxyl group at the end. So our fatty acids, they're usually made up of about 16 to 18 carbons, but it can vary. And the fact that we have this carboxyl group at the end, it's actually going to give fatty acids their acidic properties. And these long hydrocarbon chains, they are hydrophobic because of the abundance of these nonpolar carbon to hydrogen bonds. And these fat molecules, they are formed when we have 3 fatty acids that come together with 1 glycerol. And the way that they come together is through ester linkages. So our ester linkages, they bond glycerol and fatty acids. And the way that this occurs is through dehydration reaction. So when we have 3 fatty acids join 1 glycerol, we get something called a triacylglycerol. And another word for triacylglycerol is actually triglyceride, and another name for that is fat. So the 3 fatty acids that come together, they can be identical or different, and the fatty acids, depending on their structure, that's actually going to contribute to the molecule's diversity. So we can have different kinds of fats, and the fats can be saturated or unsaturated. So we know that our fatty acids, they vary in length, but they can also vary in carbon to carbon double bonds. And the location, the absence or presence of these double bonds, that's going to result in either a saturated or unsaturated fatty acid. So when we have a saturated fatty acid, we don't have any double bonds. There are no carbon-to-carbon double bonds. That is going to allow for maximum hydrogen bonding, and it is going to form straight chains. And these straight chains that we see here in our saturated fatty acids, they are going to, these straight chains, they are going to enable tight packing. And that's exactly why we have these saturated fats that are solid at room temperature, such as butter, because they have tightly packed straight chains. And these saturated fats, they're normally found in animal fats. But on the other hand, we have unsaturated fats. These unsaturated fatty acids, they're always going to contain at least one carbon-to-carbon double bond, at least one. And this carbon-to-carbon double bond, it's going to form a kink in our chain. And normally, we see these carbon-to-carbon double bonds in a cis configuration. And remember, what I mean by cis is that the hydrogen that is attached to the carbon, they're found on the same side. So this is a cis unsaturated fatty acid. But we can also have trans unsaturated fatty acids. And what that would look like is like this. We have our carbon-to-carbon double bond, we have a hydrogen, and then we have another hydrogen on the opposite sides. Remember, cis configuration always means that the hydrogens are on the same side, and a trans configuration is going to mean that it's on different sides. So basically, what happens when we have these carbon-to-carbon double bonds is that our chain is no longer straight. It's going to be kinked. And this kink, this bend in the chain, it is going to stop it from being able to be tightly packed, which is why our unsaturated fatty acids, our unsaturated fats, they are liquid at room temperature. And that includes our plant and fish oils, such as olive oil. And the way that we obtain trans fats from our unsaturated fatty acids is through a process that is called hydrogenation. So hydrogenation occurs when unsaturated fats are converted to saturated fats. And this process, it can also produce trans double bonds. And these trans fats, they negatively affect our human health. And it leads us to have a greater risk of diseases such as cardiovascular disease. So we talk about our saturated or unsaturated fats and our trans fats in terms of nutrition. So when we talk about saturated fats, they can lead to atherosclerosis. And atherosclerosis is plaque that builds up in our arteries. And then trans fats, they can exacerbate those effects. And we have hydrogenated vegetable oils, and those are found in products like peanut butter and margarine. And this hydrogenated vegetable oils, that's going to stop the separation of our lipids as oil. And then we also have essential fatty acids. And essential fatty acids, they are specific unsaturated fats. So we have essential ones as well, which are specific unsaturated fats, and they include omega-3 fatty acids. And omega-3 fatty acids, they are necessary. They are vital for our health, but they are essential because we cannot synthesize them within ourselves. These omega-3 fatty acids, they have to come from our diet. So fats have a lot of different functions, but their primary function is energy storage. So fats store energy. And fats, they actually hold 2 times the energy of a polysaccharide like starch. So if you guys remember, plants, they store energy as starch, and when they need it, they're going to hydrolyze starch to create glucose, which is an active energy form for them. But as plants store energy as bulky starch, they also have oils that are used in seeds for compact storage during dispersal. And this is necessary for plants, for them to be able to reproduce properly. And animals rely on fats also. And these fats for animals, they are portable long-term energy reserves, which can be found in our adipose cells. So our adipose tissues store our fats, and these adipose cells, they actually are going to swell and shrink as fat is either deposited or withdrawn. And fats, they also cushion our organs, such as our kidneys, and they even provide insulation. Especially we see fat used as insulation in marine mammals like whales or seals. They have blubber, which is just a thick fat layer, a thick subcutaneous fat layer that is going to allow them to retain heat. So another form of lipids, something else about lipids, is that they create fatty acids, and they create phospholipids. Sorry, and phospholipids, they are the foundation of cellular membranes. For example, how do I say? Phospholipids, they are completely essential for life. They are the major constituents of our cell membranes. And these phospholipids, they are the perfect example of what I mean when I say that form follows function. The form of these phospholipids is going to determine the function that they have. And this form follows function. Their structure, it allows them to actually self-assemble into a bilayer that is selective to our cells. So our phospholipids, they have a structure of two fatty acid tails, which is what we see here. And these fatty acid tails, they are going to be attached to our glycerol. And our glycerol, it has a third position, which is what we see here. And this third position is going to be occupied by a phosphate group. And this phosphate group is actually hydrophilic. That means that it loves water. And this phosphate group actually has a negative charge. And connected to our phosphate group, we have an R group. And R groups, they are just usually small. They are normally polar or charged, and they attach to the phosphate group, and they contribute to the variety of phospholipids that we have. So when phospholipids are exposed to water, they form bilayers. And what happens in these bilayers is that we have our hydrophobic heads, which is what we see here, and it faces out to the water. So here we just have a whole bunch of water molecules that are interacting with our hydrophilic head. And then we have our hydrophobic tails, and they face inward. So we actually, when we have a phospholipid bilayer, we have two fatty acids. One of them is always going to have a kink. And then we're going to have them be structured like this. The two hydrophobic tails, they are near each other. And the hydrophilic heads, they are going to be surrounded by water. And this arrangement, it's going to form this phospholipid bilayer, which is the structural basis of all of our cellular membranes. It separates our internal cellular environment from the external one, and it's regulating the passage of substances in and out of the cell. So some lipids can be categorized as steroids. And steroids, are characterized by a carbon skeleton that has 4 fused rings. And the specific functions of these steroids, it depends on the functional groups that they have. So cholesterol, it is a steroid. And cholesterol is extremely important, and it serves as a component of animal cell membranes. And it's actually a precursor for other steroids, including our sex hormones, such as testosterone and estradiol. So, while cholesterol, it is extremely important when we have excess cholesterol, when we have high levels in the blood, it can actually lead to cardiovascular diseases. And both saturated and trans fats, they're going to negatively affect our cholesterol levels, which increase the risk of of atherosclerosis. And when we look here, we have the skeleton of a cholesterol. And then what we see here, like here we have all these different functional groups. And this is what a cholesterol steroid looks like. So our steroids, they differ in their functional groups. Their functional groups is what's going to give them their function. And while cholesterol is extremely important, if we have too much of it, it's going to give us health issues. So overall, lipids, they are indispensable, and they are extremely diverse in their forms and in their functions. We have fats, right? So we have lipids and we have fats. And fats, they're important for energy storage. They're important for cushioning us, our organs, and they're important for insulating us. Another kind of lipid that we have are our phospholipids. And they form the structural framework of cell membranes. And the last lipid that we went over is steroids, which includes cholesterol, and it functions in membrane structure and hormone signaling. So from lipids being hydrophobic to the different biological roles that they have, they're able to demonstrate so much adaptability in our molecules that it's going to support the processes of life. So the next section that we're going to talk about is our proteins, which is another macromolecule. And proteins, they have an extreme diversity of structures. And these differences in their structures, it's going to result in a wide range of functions. So proteins, they include a diversity of structures, and these proteins, they're going to account for more than 50% of a cell's dry mass. They are so instrumental in almost everything that an organism does. And we have different kinds of proteins, and they all do different roles. So we have that they include structural support, storage, they include transportation, cellular communication, movement, and they even defend against foreign substances. But most importantly, our proteins, they include enzymes. And these enzymes, they function as catalysts in our cells, and they regulate our metabolism by selectively accelerating chemical reactions without being consumed. And because enzymes, because they can perform their functions over and over again without being consumed, we can think of them like the workhorses that are going to keep the processes of life going. So really, without proteins, life would not be possible. So proteins are really diverse in their structure, and we have Us humans, we have tens of thousands of proteins that are all different, and each of them have a unique structure and function. Proteins, they are extremely structurally complex, they are extremely sophisticated, and every single type of protein is going to have a three-dimensional shape. So, protein structure is so important that there is an entire field of study that is going to dedicate itself to protein structure. And protein structure is constantly being studied, and we can actually picture their structure using something called X-ray crystallography. And myoglobin was actually the first protein to ever have its 3D structure known. And we find myoglobin in our muscles. So, for example, in our heart and muscle and skeletal muscles, we find myoglobin a lot. And as diverse as all proteins are, they are unbranched polymers that are made up of 20 amino acid monomers. So we have only 20 amino acids that are going to construct all of our proteins. So from these 20 amino acids, we make all of the thousands and thousands of proteins that we have. And polymers of amino acids, they're called polypeptides, which is what we see here in this chain. We see a lot of different amino acids. Here we see the three-letter codes of our amino acids, which includes cysteine, leucine, and so on. And all of these amino acids coming together, they're going to be called polypeptides. So polymers of amino acids, they are called polypeptides. And biologically, a protein is a functional molecule that is going to consist of one or more polypeptides that are folded and coiled into a specific conformation. So amino acids are actually the building blocks of proteins. Like we've talked about, we have 20 amino acids, and all of our proteins are made up of the same 20 amino acids. And the bonds that we find between our amino acids are called peptide bonds. And to define an amino acid, they are organic molecules that have carboxyl and amino groups. And at the center of an amino acid, and which is what we see here, is just the general structure of an amino acid. But at the center, we have an asymmetric carbon, which is called the alpha carbon. And there are four components that attach itself to the alpha carbon. We have a hydrogen atom, we have our carboxyl group, we have an amino group, and we have a variable R group, or also referred to as a side chain. And the different R groups that we have are going to determine the amino acid properties. It's going to characterize the different amino acids. And the R group, it can be so simple as a hydrogen atom, which is going to be seen in glycine, or it can even be a carbon skeleton that has a variety of different functional groups, which is something that we see in the amino acid glutamine. And the R group is going to determine if the amino acid is polar, nonpolar, polar, acidic, or basic. So the physical and chemical properties of our amino acids, they're determined by our R group. So our R group is going to determine the unique characteristics of a particular amino acid. So one group of amino acids, it has nonpolar R groups, which make it hydrophobic, which is what we see here. Glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline, they are all nonpolar hydrophobic amino acids. Now we also have a hydrophilic polar amino acid. And the difference between hydrophobic and hydrophilic amino acids, again, is their R groups. And then we have another group of amino acids, which has functional groups that are ionized. They are charged at the cellular pH. And some of these R groups, they have a negative charge because they have an extra carboxyl group, which is what we see here. But basic R groups have amino groups that are positively charged, which is what we're seeing here. Basic R groups have amino groups with a positive charge. But every single amino acid is going to have a carboxyl group and an amino group. And the terms acidic and basic in the context of amino acids is going to refer only to the R groups. So going into more detail of our peptide bonds and our polypeptides, we have already talked about amino acids being joined together, and it is joined through something called a peptide bond. And these peptide bonds, they result from dehydration reaction. So the dehydration reaction, it is going to remove a hydroxyl group from the carboxyl end, which is over here, our hydroxyl group, also OH. It is going to remove that hydroxyl group from the carboxyl end of the amino acid, and it's going to remove the hydrogen atom from a different amino group of another amino acid. And this removal is going to cause water to be lost, and it's going to form our peptide bond. And when we have this process be repeated over and over again, we're going to create our polypeptide chain. And at one end of this polypeptide chain, which is what we're seeing here, we have a amino acid that has a free amino group, which is what we see here. It is completely free, and it is called the N-terminus. But at the other end of our amino acid, we have a free carboxyl group, which is known as the C-terminus. So polypeptides, they range in sizes from a few monomers to even thousands. And each polypeptide is going to have a unique linear sequence of amino acids. And this unique sequence is going to define our polypeptides. So now we're going to talk about protein conformation and function. And again, proteins are a perfect example of form follows function. So their shape is going to directly determine the role that they have in the body. So we can have two different protein shapes. Well, these are the two main shapes. We can have more. We have fibrous proteins, which are elongated. They are also insoluble. And this structure makes them really well suited for structural functions. We can find fibrous proteins in places like collagen. And collagen is going to give strength to connective tissues. And we can even find these fibrous proteins in keratin. And keratin is found in our nails and in our hair. And another form of structure that we see proteins have is globular. And globular proteins, as you can see, they are compact, they are spherical, they normally are involved in things such as transport and regulation. Examples of them include enzymes, hemoglobin, and antibodies. So protein function is going to depend on the precise three-dimensional shape that it has. And we know that our proteins, they are made up of polypeptides. And a polypeptide chain is a straight chain. And this polypeptide chain, it's actually going to spontaneously fold. And when it folds, it's going to take a three-dimensional structure. And this folding process, it's driven by amino acids in the chain. And then it's reinforced by the bonds that it has, which include hydrogen and ionic bonds. It includes van der Waals interactions that are found between different parts of the chain. And because structure determines function, its ability to interact with other molecules is so important. So for example, we have antibodies, and antibodies, they're going to recognize and bind to specific foreign substances that are going to help in our immune response. We have endorphins. Endorphins, they bind to receptors in our brain. They relieve pain and they give us that feeling of euphoria. And then we have drugs like morphine and heroin even, that it's going to mimic the shape of endorphins, and this mimicry is going to allow them to bind to the same receptors that our natural endorphins bind to, and it's going to produce similar effects. So in almost every single case, a protein's ability to function is going to depend on its shape. And proteins, when they misfold, they often lose their functionality, and that can lead to severe consequences and diseases. So the function of a protein is an emergent property that results from its specific molecular order. In other words, the structure of a protein is hierarchical, with 4 levels contributing to its final shape and function. So the primary structure of a protein, it is the unique sequence of amino acids that is found within a polypeptide chain. And this sequence is determined by genetic information, and it's going to dictate how it's going to fold. So the sequence is determined by genetic information, and this determines how the protein is going to fold. And when we have even a small change in the primary structure of this protein, for example, if a single amino acid is substituted by another one, it can have a significant impact on the function, which, for example, we have sickle cell disease. And what happens in sickle cell disease is that glutamic acid is going to replace valine inside of our hemoglobin, which is going to lead to the misfolding and improper function of our blood cells. They're going to be unable to carry of hemoglobin in our blood cells, which means it's going to be unable to carry oxygen normally, and it's going to have other associated consequences as well. So this is what the primary structure of our proteins look like. It is our polypeptide chains. It is a sort of 2D structure, okay? Sort of. So the secondary structure of proteins that we have, it involves regular coils and folds in our peptide chains, which is what we see here. This coiling of our polypeptide chain. And this coiling is actually going to be stabilized by hydrogen bonds. And we have 2 common secondary structures. We have alpha helixes, and alpha helices, they are delicate coils that are held together by hydrogen bonds, because hydrogen bonds, they stabilize this structure between the backbone's repeating units. So another form of the secondary structure that we have are beta-pleated sheets. And these beta-pleated sheets, they are flat and folded structures that form when segments of this polypeptide chain are aligned and connected by hydrogen bonds. And these beta-pleated sheets, they're found in proteins like silk, where the numerous hydrogen bonds make that material really strong. So the tertiary structure of a protein, that is actually the overall dimensional shape of a single polypeptide chain. So it is of a single polypeptide chain. And this is going to result from the interaction of R groups and of the amino acids. So for example, we have hydrogen bonds that are found between polar groups Ionic bonds are found between charged side chains, are hydrophobic interactions, and van der Waals forces, they cluster the nonpolar side chains inwards. And then we have disulfide bridges, and these disulfide bridges, they're actually covalent bonds. And these are covalent bonds that occur between sulfur atoms and cysteine residues that are going to provide additional stability to this. And the last structure is a quaternary structure. So some proteins, they're composed of 2 or more polypeptide chains, and they come together to form a functional protein. And examples of this include hemoglobin and collagen. And like I said, hemoglobin is globular and collagen is fibrous. And both of these are actually quaternary structures of proteins. And a hemoglobin, which is a globular protein, it's going to have 4 subunits, 2 alpha and 2 beta chains, and it's going to have a heme group that binds oxygen. And collagens, they're fibrous proteins. They have 3 polypeptide chains that are coiled into a triple helix, and they're going to provide structural strength to connective tissues. So, protein folding and denaturation is extremely important. And this is because proteins, they have to fold correctly in order to function. So the protein's structure also depends on the physical and chemical conditions of the protein's environment. So protein folding is usually assisted by chaperones, and it's going to protect the polypeptide chain while it folds. It's going to protect from alterations in the normal environment. So if we have abnormal pH, if we have abnormal temperature, these chaperones are going to protect the protein from these aberrations of appropriate internal cellular conditions. And when we have improper folding, that's actually going to lead to diseases such as Alzheimer's and Parkinson's. It can lead to even mad cow disease. And the environmental factors such as pH or temperature, even salt concentration, that all can affect a protein. It can disrupt its structure, and it's going to denature it, and it's going to lose its function. So for example, when we have high fevers, that can denature the proteins in our blood, and that's fatal to us. When we are transferring a protein to a nonpolar solvent, that can also cause it to refold with hydrophobic regions exposed, which are going to make it be inactive. And some proteins, although rare and it can't always happen, some proteins are able to refold into their functional shape after it is denatured. And this tells us that the primary structure of our proteins, it is going to hold all of the information that a protein needs in order to fold properly. So, there have been a lot of advances in protein research, and modern techniques like X-ray crystallography, which is what discovered myoglobin structure, bioinformatics, NMR spectroscopy, they all have advanced our understanding of protein structure. X-ray crystallography, it actually is going to determine the three-dimensional structure of a protein, but it requires a protein to be crystallized, and that crystallization can lead to aberrations in its structure or the way that we see things. So we have 2 different options, which are nuclear magnetic resonance, NMR spectroscopy, and we also have bioinformatics. So NMR spectroscopy, it's not going to require the crystallization of proteins, but it's also used to determine protein structure. In bioinformatics is going to use computer programs, is going to use computational tools, and that is what's going to be able to predict that three-dimensional structure of polypeptides from their amino acid sequences. So understanding protein conformation and its structure, it's extremely important for exploring their function and their roles in health and disease. So when we're looking at this picture, this is a picture of an electron density map of protein structure that was determined by X-ray crystallography. And what this does is that the blue meshes that we see, it is representing those regions that have a really high electron density. And the atoms, they are located in a protein structure. So that is what the blue mesh is representing, where the atoms are located in a protein structure. And then we have yellow lines, and those yellow lines, they're going to depict the modeled polypeptide backbone fitted somewhere into that electron density, which is going to come together to reveal the shape, the 3D shape of the protein. And then we have specific atoms, and we have also the side chains, which they're going to be highlighted in pink and red. And those are also modeled to match the density for accurate structural determination. So the last section of macromolecules that we're going to talk about is nucleic acids. And nucleic acids are so important because they store, transmit, and help to express our hereditary information. So genes, we've previously described them as the unit of inheritance. And genes, they are going to program our amino acid sequences of polypeptides. So a gene, it consists of DNA, which is a polymer that is known as nucleic acid. And nucleic acids, they are monomers that are made up of nucleotides. And we have two different types of nucleic acids. We have RNA, which is ribonucleic acid, and we have DNA, which is deoxyribonucleic acid. So DNA is a double-stranded molecule that forms a double helix shape. And DNA, the sugar that is found in it is deoxyribose, and it is able to elicit base pairing where we have adenine and thymine and then guanine and cytosine that pair together. The function of DNA is to store genetic information that is passed from one generation to another, and it's actually going to direct its own replication, and it's going to indirectly modulate the synthesis of protein through RNA. And this process is called gene expression. DNA is found in eukaryotic cells in the nucleus, and it's also found in prokaryotic cells, but it is found in the cytoplasm. And we have our RNA, which is single-stranded, though there is one type of RNA that is able to form base pairs, and we're going to go over that later. The sugar that is found in RNA is called ribose, and it follows the same base pairing rules as DNA, except in RNA, instead of thymine, we have uracil, but thymine is going to pair with adenine, and guanine is going to pair with cytosine. RNA is an intermediary of gene expression, and we have different types of RNA. We have messenger RNA, which is going to carry instructions for protein synthesis from DNA to the ribosomes. We're going to have transfer RNA, which is what is able to form base pairs, and transfer RNA is going to bring amino acids to the ribosome during protein synthesis. And then we have rRNA, or ribosomal RNA, which is a structural component of ribosomes. So RNA is actually able to exist in different shapes, and it's essential for the cell's machinery to create proteins. So the inheritance and function of DNA. Well, organisms, they are going to inherit their DNA from their parents. And before cell division, right? So before cell division, our DNA is going to replicate. And this replication prior to the division of cells is what's going to ensure that the genetic information that is passed to the daughter cells is identical. It's an identical copy of the genetic information of DNA. And DNA provides instructions that is going to produce proteins and other cellular components. And that's actually called gene expression, the process of converting information from a gene into a functional product, which is usually a protein. And that flow is always going to occur from in DNA makes RNA, which makes protein, which is our central dogma of biology. And that's always going to be true. So each gene that is found along a DNA molecule, that is what's going to synthesize, it's what's going to direct the synthesis of a specific type of RNA, which is called mRNA, messenger RNA. And then that mRNA molecule is going to interact with the cell's protein synthesizing machinery, which are known as our ribosomes. And that is what's going to determine the order of amino acids in a polypeptide. So when we're thinking of DNA and RNA in the cellular context, we have a few key differences, which is all in this table. And what we see is that in eukaryotes, our DNA is found within the nucleus. The ribosomes, which are the location of protein synthesis, they are found in the cytoplasm. And mRNA, its function is as an intermediary, which carries genetic instructions from the nucleus to the cytoplasm. But in prokaryotes, since prokaryotes don't have a nucleus and they do not have membrane-bound organelles, they have the location of their DNA and RNA in the same spot, which is the cytoplasm. And the function of mRNA is the same in the sense of it's going to carry our genetic information to our ribosomes. So the structure of nucleic acids, that structure is a polymer that is made up of nucleotide monomers, which are organized as polynucleotides. And each nucleotide, it is going to consist of 3 parts. It's going to have a nitrogenous base, a pentose sugar, and a phosphate group. And our nitrogenous bases, we have 2 different ones. We have pyrimidines, which are single-ringed structures, and they include our cytosine, uracil, and thymine. And it's important for us to know that thymine is only found in DNA, and uracil is only found in RNA. And the next nitrogenous base that we have is purines, which include our adenine and our guanine. So the pentose sugar that we have, in RNA, we have ribose, and we have deoxyribose in DNA. And the way that these sugars differ is that deoxyribose is not going to have an oxygen on its second carbon, oxygen atom on carbon 2. And our ribose is going to have an oxygen on our second carbon. And the last component of a nucleotide is the phosphate group. And this phosphate group is going to attach to the sugar molecule, and it's going to link nucleotides in a chain. So our phosphate group is added to a nucleoside, which is defined as our sugar and base, which is going to form a nucleotide. So again, nucleosides, they consist of sugar and a nitrogenous base. But nucleotides, they are going to consist of nucleoside plus 1 or more phosphate groups. So polynucleotides, They are formed by the linking of adjacent nucleotides through phosphodiester bonds. And these phosphodiester linkages, they form between the hydroxyl end and the hydroxyl end on the 3' carbon of one nucleotide and the phosphate on the 5' carbon of the next nucleotide. And this is going to result in a sugar phosphate backbone that has nitrogenous bases as side chains. And the directionality of our polynucleotides is really important because the two ends of polynucleotides are distinct. One end is going to have a 5' phosphate group, which is known as the 5' end, and the other one is going to have the 3' end, which is the hydroxyl group. And that sequence of bases, the sequence of our nitrogenous bases that are along a DNA or mRNA polymer, it's unique to each gene. And the order of bases in a gene is what's going to determine the sequence of amino acids in a protein. So when it comes to DNA and replication, our DNA is a double-stranded helix that has a sugar phosphate backbone on the outside that has paired nitrogenous bases on the insides. So DNA, it consists of two polynucleotide strands, and those strands, they spiral around an imaginary axis, which is going to form our double helix. And those strands, they are anti-parallel to each other. They run in opposite 5' to 3' directions. So when we put our double helix into a ladder configuration, we see that our strands are anti-parallel. Here we have 3', here we have 5'. And then at the end of our 5' we have a 3', and at the end of our 3' we have a 5' end. So base pairing rules in DNA are always going to be the same. Adenine and thymine will always pair, and guanine and cytosine will always pair. But when it comes to the replication, what happens is that before cell division, each strand of this double helix is going to serve as a template, and that template is going to create a complementary strand. And that complementary base pairing, that's what's going to ensure that the genetic information is accurately replicated. And this process, it results in two identical copies of that original DNA molecule that is going to then be distributed to daughter cells during cell division. So the complementary base pairing, it allows the sequences of one strand to dictate the sequence of the other. So when we look at the structure and function of RNA, what we know is that RNA is normally single-stranded, but that it can form base-paired regions. And the base pairing in RNA is that adenine and uracil pair, and uracil is the equivalent of thymine in DNA, but found in RNA. And then we have guanine and cytosine that pair. And the function of RNA is protein synthesis. And that's where we find out that we have different types of RNA. We have mRNA, which is going to transcribe. mRNA transcribes genetic information from DNA to ribosomes for protein synthesis. We have tRNA, transfer RNA, which is going to form base pairing, and this transfer RNA is going to transfer the amino acids to the ribosome for polypeptide formation. And then we actually have one more type of RNA, which is rRNA. And our RNA is known as ribosomal RNA, and it is an integral component of ribosomes that aid in protein synthesis. And RNA is extremely versatile because the ability that RNA has to fold into diverse shapes is what's going to allow it to perform all of its various functions. And these functions include catalyzing reactions, it aids in protein synthesis, and it serves as an intermediary between DNA and protein. So, the last section of this chapter is genomics and proteomics have transformed biological inquiry and applications. So the study of genomics and proteomics, that has completely revolutionized the field of biology. And what it did is it provided tools and techniques that allowed us to analyze and understand biological systems at a molecular level. And these advancements, they deepened our understanding of the genetic and protein sequences, which transformed how we addressed biological questions and applications. So, DNA, the structure of DNA, this was a big milestone for us. And this structure, it was first described in 1952 as a double helix. And this discovery, it laid the foundation for understanding how the linear sequences of nucleotide bases specified the amino acid sequences of our proteins. And by the 1970s, we had the first chemical techniques for DNA sequencing being developed, which is what paved the way for future innovations. Something else really important that happened was the Human Genome Project, which occurred between 1990 and the 2000s. And what happens is that the entire human genome, it was sequenced. And all of our genomes, all of our genes, it led us to understanding their functions and the genetic variations that are found in them. Since then, we have had more advances that have led to faster and more cost-effective ways that we can sequence genomes, and that actually it allowed us to sequence the genome of different organisms aside from humans. So we've actually sequenced dogs, cow, pig, chimpanzees, and zebrafish. We've even sequenced the genome of bacteria and microorganisms such as Mycobacterium tuberculosis and HIV. And we have also sequenced the genome of E. coli, Escherichia E. coli, and Drosophila melanogaster. So something else that was important is the emergence of bioinformatics. And bioinformatics is defined as the integration of computer science, statistics, and biology. In bioinformatics, it's able to manage and analyze large data sets. So the vast amount of sequencing data, it gave rise to bioinformatics. And the bioinformatics, it's a tool that enables researchers to be able to identify gene functions. It can compare genomic sequences, and it can even predict revolutionary relationships. So, moving on to genomics and proteomics, genomics, it's defined, and it's involves the analyzing of large sets of genes or whole genomes across different species, and this can actually answer biological questions. And similarly, proteomics, it focuses on analyzing large sets of protein sequences, which allow us to understand how protein structures and how these proteins, they're able to interact within an organism. And genomics and proteomics together, they address complex biological problems by providing complementary insights into the genetic blueprint and functional dynamics of cells, which enable a deeper understanding of disease mechanisms, biological processes, and even potential therapeutic targets. So DNA and protein, we can actually use it as sort of like tape measures of evolution. So genes, our DNA, our genes and their products, the products which are proteins, they serve as records of the hereditary background of an organism. And DNA, we know that it's inherited from parent to offspring, which is why siblings, they share greater DNA and protein similarity compared to unrelated individuals of the same species. And this principle, it extends to relationships between species, which are forming the basis of molecular genealogy. Molecular genealogy. And this molecular genealogy, it's just the idea that species that are closely related, based on fossil and molecular evidence, they exhibit greater similarity in their DNA and protein sequences than species that are more distantly related. So, for example, we had scientists that compared 146 amino acids in the polypeptide chain of hemoglobin to sequences in 5 other vertebrae. And what we found was that humans and gorillas, the hemoglobin, the amino acid sequence of hemoglobin, it differed by only one amino acid. So it's reflecting the idea that close genetic and functional similarity between those two species. When they did the differences and they found the differences in humans versus frogs, there was 67 amino acids, which is indicating that we have more evolutionary divergence and adaptation of hemoglobin that is specific to our different physiological needs. And despite all of the difference, all species, they maintain functional hemoglobin, which is emphasizing the idea of conservation and divergence of molecular structures over evolutionary time. So for the future, genomics and proteomics, they have applications in so many fields. When we think about it in medicine, it's able to identify genetic mutations that have been linked to diseases and and it's been able to develop personalized treatments. We can think about it in the terms of agriculture, in which engineered crops with improved traits, such as drought resistance and higher yield. We can think about it in the context of evolutionary biology, in which tracing the evolutionary history of species, and it allowed us to understand biodiversity. And continued advancements in sequencing technologies and these computational tools, they're going to further promise more breakthroughs in understanding our life at molecular levels.